Today we are looking at the lock and key model of enzyme action. On the screen we have our enzyme which acts like the lock. It has a specifically shaped area called the active site. Our substrate, the yellow triangle here, acts like the key. Notice how the shape of the substrate is perfectly complementary to the active site. As the reaction begins, the substrate binds to the active site. Because the shapes fit together so precisely, they form what we call the enzyme substrate complex. This is the critical moment where the enzymes start to work on the substrate's chemical bonds. The enzyme then catalyzes the reaction, breaking the substrate apart. You can see here that a single triangle has been converted into two separate products. Even though the substrate has changed, notice that the enzyme itself remains exactly the same shape it was in the beginning. Finally, the products are released from the active site. The enzyme is now empty and unchanged, meaning it is ready to find a new substrate and start the whole process all over again. And that is the lock and key model. Now we are looking at the induced fit model. Notice that the enzyme's active site is not a perfect match for the substrate yet. It's a bit of loose fit. As the substrate enters, the enzyme actually changes its shape to wrap tightly around it, like this. This induced fit grips the substrate perfectly to start the reaction. This chemical reaction happens and our substrate is converted into two separate products. The enzyme has done its job of catalyzing the reaction. Finally, the products are released. The enzyme relaxes back to its original shape. Look carefully. The products are released and the enzyme relaxes back to its original shape, ready to bind with a new substrate and start the process again. To understand denaturation, we first look at a normal enzyme. As we have seen, the substrate and the active site fit together perfectly. This specific shape is what allows the enzyme to do its job and catalyze reaction. However, enzymes are sensitive. If the temperature increases too much or if the pH levels fluctuate, the enzyme undergoes denaturation like this. The enzyme undergoes denaturation. You can see here that the active site is losing its shape. This protein is literally unfolding or wrapping because of the environmental stress. Once the enzyme is denatured, it's permanently damaged. Even though the substrate is still there, it can no longer fit into the destroyed active site. There is no reaction and the enzyme becomes non-functional. This is why maintaining a stable internal environment is crucial for life.